Hello everybody and welcome to Cellix's webinar on using Cellix's Vena Biochips with syringe pumps. My name is Biochips and I'm glad you can join us today. The quick agenda for today is I'll start with introduction of Cellix's Biochips and then proceed to a detailed explanation on how to use Cellix's Biochips with syringe pumps and finish with the summary. Cellix's VenaFlux platform provides complete integrated tool mimicking physiological flow and shear stress. It can mimic venous and arterial flow and visualize cell rolling and cell adhesion under shear stresses. Very similar to intravital microscopy, it can provide a functional assay where activation of cell receptors can be observed and quantified. The first in line of Cellix's biochips is Vena 8 biochip. It has 8 microcapillaries, which have a very low volume, 1 microliter per channel. It can be used with bright field and phase contrast microscopy and different magnifications from 20x to 60x and under very high shear stresses, up to 1000 dynes per square centimeters. The applications of this biochip include cell adhesion and rolling under shear stress, thrombosis, and cell proliferation. If it is necessary to use fluorescent microscopy and it is necessary to have a low fluorescent background, we recommend Vena 8 Fluoro Plus biochip. It has improved optical clarity and can be used with high numerical aperture objectives, high magnification up to 60x and 100x oil immersion, bright field phase contrast and fluorescent microscopy. It also has a plastic which is matching refractive index of glass very closely, so it can be used with confocal microscopy. So application examples, it's a low-level receptor staining of leukocytes and other cells, and also confocal imaging of different cells. In this example, you can see confocal images of endothelial cells stained with different dyes. If it is necessary to look at the cell-to-cell interaction, we recommend Vena 8 Endothelial Plus biochip, where low number of endothelial or epithelial cells can be seeded, providing the uniform and confluent layers. These layers can be pre-treated with compounds and by the shear stress. The biochip itself can be used with bright field phase and fluorescence. The applications of the biochip include endothelial cell culture, cell adhesion and rolling under flow, cell-to-cell interaction where leukocytes can interact with endothelial layer, thrombosis, cell proliferation, or cell staining. Another chip in line of Vena biochips is Vena EC biochips. It has ability to use different substrate materials because the chip is separated into parts, the substrate and the cover, which has 2 channels. It can be used with bright field phase fluorescent microscopy, 20 to 40 long working distance objectives, and has a very low fluorescent background. It is also possible to form the confluent layers of endothelial epithelial cells on the substrate using conventional incubator and standard 6-well plates. The applications of the biochips include cell-to-cell interaction, endothelial epithelial cell culture, cell rolling, and adhesion under flow. The key components of every flow system is as follows. It's the pump, typically it's syringe pump, microscope to visualize the microcapillary or flow chamber, camera to acquire the images, and analysis software. Cellix provides 2 types of the system, custom-built systems which build around your existing microscopes, and also we provide fully integrated system, VenaFlux platform, which integrates all these components in one system. Myers nanopump, which is ultra-precision pumps, and covers a very big range of shear stresses from very low 0.05 dynes per square centimeters to very high 1000 dynes per square centimeter, typically used for thrombosis experiments. The flow rate is precisely controlled via flow sensors, and this pump allows parallel perfusions with attachment of multi-flow aid. The flow direction is fully reversible and shear stress is controlled with high accuracy. The Myers nanopump runs together with VenaFlux assay software, and this is fully computer controlled pump. Cellix's biochips can also be used with standard syringe pumps, like syringe pumps from Harvard apparatus or World Precision Instruments or KD Scientific pumps. I'll give you two examples of cell assays which can be carried out using Cellix's biochips. First is the leukocyte adhesion assay. It can be run using this Myers syringe pump because of the low shear stress required. You can coat the channels with a range of different ligands, VCAM1, ICAM1, selectin, fibronectin, and cells can be different leukocytes, THP1, PVMCs, or neutrophils. Typically, we use 2 to 5 million cells per milliliter concentration of cells. Shear stress varies from 0.5 dyne to 2 dynes per square centimeter, which corresponds to volume type of the assay, which is possible to carry out using Cellix's biochip, is thrombosis assay. This can be run using Myers nanopump and also conventional syringe pump. Typical channel coating is collagen, von Willebrand factor, or fibrinogen. In this case, we use whole blood sample, and shear stresses varies from 22 dynes to 225 dynes, which corresponds to shear rate of 500 to 5,000 inverted seconds. And typical volume used in this assay is from 20 microliters to 200 microliters. Before running the experiment, the biochips needs to be coated with ligand. You can do it using just a standard pipette, and it requires 10 microliter of ligands, which is just pipetted in each individual channels. The biochips is then stored at 4 degrees overnight in humidified conditions. Just prior to experiment, half an hour prior to experiment, you can block the biochip with PSA to avoid any non-specific adhesion. Cellix's biochips can be used with the aid of Myers nanopump. In this case, the pump is connected to biochip using special 8-way connector. And this is the attachment to a pump, which is called multi-flow aid, which, if it is required to have a high throughput of your experiments, Myers nanopump can run 8 channels simultaneously. So, to do the flow experiment, you need to insert the cable of the Myers nanopump into the biochip and to wash any excess of your unbound ligand by dispensing into the output wells. Then manually or with the aid of robotic dispenser, the system can pick up and dispose the waste. Following that, the system will place the sample into each of the individual wells, and the sample will be pulled through the capillaries at the specified shear stress and then can be absorbed on the microscope. If it is necessary to run the Cellix's biochip with conventional pumps, we provide a separate single-way connector, which has a little pin on one side and the lure connector on the other side. This can be plugged into a standard syringe or any syringe on the syringe pump. If this cable is not long enough, we provide the extension, which can extend it up to 50 centimeters. To do the biochip assay, you first connect your cable to the syringe pump and then wet the tip of the connector cable. Insert it into biochip and wash any excess of the ligand. Then following that, you dispose the waste with pipette and place the sample instant into the input valves. The sample is then pulled through the biochip and the interaction of cells with the biochip coated walls observed. If the sample is quite large, more than the well can hold, then you can use the extension cable and we can be pulled from the separate Eppendorf. Cellix's biochips can be used in a variety of different applications: in inflammation for looking at leukocyte adhesion studies, cardiovascular in atherosclerosis and thrombosis, bacteriology for looking at bacterial and biofilm formation, respiratory, allergy and asthma, and also in oncology for looking at melanomas and metastasis of cancer cells. In summary, Cellix's products provide physiologically relevant tool which mimics in vivo shear stress flow